sharing my screen now. All right, we're going to be doing lab 5, streaking for isolation and culture media. You should be reading the unknown project instructions, which you can find on Canvas. I think it's week 5. Let me look there. But you should read the instructions because next week I'm going to be talking about the unknown project. Any questions about that?
Yeah, how would we find it on Canvas? Because week 5 lecture, it doesn't show anything for the unknown project. And then week 4, all it says is just read the unknown project. There's no link to it.
It should be in week 5 lab. Is that showing?
Week 5 lab, there's no lab for week 5 open. It just says lecture.
Let me check. Four lab is showing, week 5 lab is not showing, so it should be showing now. And you want to do the unknown report. And I want you to read the unknown instructions. If you have trouble with the rich text format, let me know. I know that the new Microsoft is having trouble reading rich text format, and I'll make a Word doc X if you're having trouble with that. Anyways, I want you to read this. It wouldn't hurt you. Sometime in the future, you're going to want to take a look at the grading for the unknown project. The unknown list, this is the species of bacteria. You have to decide which one yours is. So I don't know, there's like 40 different species, maybe 60 different species. You should probably look over building a dichotomous game plan. That's what the unknown project is going to be doing. And that is, you're going to be choosing one test at a time, and it can be a positive and a negative result or something similar. And that's a dichotomous game plan. So if it's positive, we then go this way. And then you do a test after that, and it too would be positive and negative. And you run another test, and let's say it's positive and positive. So it's here and then here. The point is you're supposed to be asking for one test at a time and using a dichotomous game plan. Anyways, you should read about that. And I'll talk more about it when I talk about the unknown project, which is this right here, the instructions. And then this is a sample journal. A professor wrote up the lab report and then made it available as an outline, or not as an outline, as an example of what your lab report should look like. And it's not loading here, but anyways, that one you don't need to look at right away, but you'll want to look at it like when you're writing your unknown report, you want it to look something like this. And this is taking a long time to read. Okay, any questions about any of that? So at least read the unknown project instructions file, which is on lab 5. And let me come back to that. I'm going to guess lab 6 is turned off. I turn off lab 5 because it's so big. I don't want to overwhelm the students at the first. Lab 6 is pretty short, but lab 5 is really long. Most of them you won't be doing. Looks like week 8 is all there. 9 is all there. 10 is all there. Okay. So I think everybody's turned on. On week 5, there are a whole bunch of different tests that you can run on your unknown bacteria. And obviously, you're not going to be running all of these tests. You will run a test, and then depending on what your results are, you'll look up to see what would be another good test to run, and then you'll check that test.
While you're in there, can you also open the PowerPoints too from the week, from this, or last week, sorry.
Is that week 3?
2 and 3.
Sorry, I haven't been feeling well, and so I haven't done a lot of things I should have done. It looks like chapter 2 is already open. But chapter 3 I just made available. Okay. Let's go on to the lab. So we're doing lab 5 today, cultivation of bacteria. We'll talk about different culture media, inoculation and isolation. There is some reading that you should do before beginning this lab. And there is a video clip and a virtual lab for today. I'll have to check to make sure the virtual lab still works, but hopefully there's a virtual lab. The primary learning objectives: upon successful completion of the lab, students should be able to understand the importance of proper specimen collection and transport to the lab, explain the 5 principal steps used by microbiologists to process the specimen, differentiate between the various types of culture media, explain the purpose of streak plate culture technique, AKA streaking for colony isolation, define the following terms: selective media, differential media, and reducing media. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, drying, and exposure to oxygen, anaerobes, and require special transport medium to preserve the specimen. Once the specimen is received in the lab for microbiological culture, microbiologists employ the 5 principal I steps to process it. First, inoculation, placing the specimen on artificial culture media to initiate growth. Isolation, spatially separating microbial growth into distinct colonies so that we can isolate specific species. And you probably will need to isolate it 2 times, meaning send it through colony isolation 2 times to isolate into distinct species. Incubation, placing the culture medium in the environment conditions to facilitate growth. Inspection, examine the culture for the presence of microbial growth. And then identification, performing specific tests to ID any pathogenic microbes that are isolated. The 5 steps will be described, and they take different types of culture media. You're already familiar with a general purpose media, and that would be Na or nutrient agar or NA, nutrient agar with yeast extract, or TSA, tryptic soy agar. And these bacteria promote the growth of many organisms, but they do not promote the growth of fastidious microbes that have very specific nutritional requirements. To grow fastidious microbes, we have to use an enriched general purpose media. And one enriched general purpose media is blood agar, which is, I think, tryptic soy agar, that's supplemented with 5% sheep's red blood cells. And the sheep's red blood cells provide nutrients that the fastidious organism requires, or at least some fastidious organisms require. Anyways, that's general purpose media, and then enriched general purpose media. There's also selective media. Selective media contains a chemical that inhibits or halts the growth of some organism, but does not inhibit the growth of other organisms. Keep in mind two things when using selective media. First, selective media isn't necessarily 100% perfectly selective. Second, even those organisms that are supposed to grow on the selective media may form smaller colonies than they would grow on a general purpose media. Any question about any of that? All right, there's also differential media, and it contains chemicals that produce visible differences among different groups of cells when they're grown on that media. The differences are unrelated to how well the organism can grow on the media. The differential cultures allow you to distinguish between dissimilar bacteria visually by distinguishing two different colonies from each other. And the cells usually, or the colonies, look different. They may have a different color. And lastly, there's reducing media. Reducing media helps promote the growth of anaerobic bacteria. The reducing media has a reducing compound in it, which removes oxygen. And so the reducing media will have low oxygen or no oxygen. And that is required by strict anaerobes. They are tricky to grow though, because you have to be, you can't expose the cells to oxygen at any time. So for example, like when you're moving from one culture to a next with a loop, you're exposing them to the air. And if they're really susceptible to oxygen, they would not grow after being exposed to the air. Usually they can live with brief exposure to the air, so that if you're moving just from one culture to another, it's usually not a problem. It's important to note that culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential, so we call it a selective and differential media. McConkey's agar is selective in that if something grows on the play, it is likely to be gram-negative that grows, meaning McConkey's selects against gram-positive. And why that's the case is just the chemicals in it. It has bile salts in it, which are found in the intestine. And obviously gram-negatives tend to grow in the intestine. And gram-positives don't like growing around bile salts, but something that grows in the intestine has to be around bile salts. It also has crystal violet in the media. And crystal violet gets in the gram-positive cell and then prevents the cell from growing. And that's why McConkey's auger tends to look, I don't know, purplish. It's from the crystal violet in the media. So if it grows, it's likely to be a gram-negative, but it's also selective and differential, and it differentiates between lactose fermenters and non-fermenters. If it's a lactose non-fermenter, the bacteria will appear non-pink colonies. If the bacteria ferments the sugar lactose, that lowers the pH and the colonies appear pink. So this bacteria is lactose fermenter. This one is a non-lactose fermenter, and they're both gram negatives because they're growing on McConkey's. Any question about any of that? We mentioned blood agar. It's an enriched general purpose media. And it is also, besides an enriched media, or enriched general purpose, I guess, it's also a differential media that you can differentiate cells growing on the blood agar by their hemolytic pattern. You can have cells which have gamma hemolysis, meaning no hemolysis. You can have cells which have alpha hemolysis. And you see the darkening of the media around the alpha as well as inside the alpha. The darkening is from partial hemolysis of the blood. And then you can also have beta hemolysis, and that makes complete lysis of the blood cells. And so the beta here, shown in white, is the bacteria. And then the clear regions around the cells are the beta hemolysis, that's complete hemolysis, where all of the cells are hemolyzed. You'll note that there is a ring of alpha hemolysis around the beta hemolysis. And this is typically something we see, that around beta hemolysis, further out, we have partial hemolysis. But this we call beta hemolysis. That's alpha hemolysis, and that's gamma hemolysis on a blood agar plate. Any questions about any of that? So this table shows you the different types of media, general purpose media, which includes TSA, NA, and NA. Enriched general purpose media, which includes blood agar and chocolate agar. And then the blood agar is also differential because you can distinguish differences in the hemolytic activity of the cells. So blood agar is differential and enriched. Chocolate agar is only an enriched general purpose media. The difference between chocolate agar and blood agar is we add the blood cells to the chocolate agar when the agar just came out of the autoclave. So the agar is quite hot, and that causes the red blood cells to lyse, which gives the media a brown appearance. And that's why it's called chocolate agar, because chocolate is brown. Blood agar, we take the media out of the autoclave and then let it cool to something like 50 to 55 degrees. And only when the temperature of the media is within that range that we add the blood agar. And that prevents the blood from being lysed because the agar is still fluid, but it's cool enough that it doesn't lyse the blood. And then below 50 degrees, I don't remember what temperature exactly, but somewhere below 50, either 49 or 48 or something like that, the agar starts to solidify. And so you have to add the blood while the agar is still a liquid. McConkey's agar and eosine methylene blue, also called EMB, or hectone agar, HE. These agars are all selective and differential. They're selective in that they allow gram-negative bacteria to grow, and they select against gram-positive cells. And they're differential on their ability to show lactose fermentation. And they will have different colors if they ferment the sugar lactose. McConkey's, if it ferments the sugar lactose, will be pink. Hectone HE agar, it's a salmon color if it ferments the sugar lactose. And then EMB, I think it's greenish or blackish if it ferments the sugar lactose. PEA agar, or phenyl ethyl alcohol, is a selective media, and it is used to isolate gram-positive bacteria. So phenyl ethyl alcohol grows gram-positive bacteria. Gram-negative do not tend to grow in it, and that's because the alcohol, it's in the gram-negative cell wall and prevents those cells from growing. Mannitol salt agar is a selective and differential media. It's differential on the basis of mannitol fermentation. If the bacteria ferment the sugar mannitol, the bacteria will have a yellow halo around the colony. The colony may look yellow, but it may not. But it'll have a yellow halo around the colony. We can use this to separate Staphylococcus aureus from other Staphylococcus species, because Staphylococcus aureus ferments the sugar mannitol, and other Staphylococcal species do not. So Staphylococcus aureus will have a yellow halo, and other Staphylococcus species do not have a yellow halo. A mannitol salt agar is also selective, and if the bacteria can grow on the plate, that means they like growing around salt. A mannitol salt agar has 7% sodium chloride in it, and most bacteria will not grow above 1 to 2% salt, and this has 7% salt in it. And mannitol salt is something I may ask you what is in it, because it tells you in the name what's in it. It has salt in it, sodium chloride, 7%, and it has the sugar mannitol in it. Okay?
So
Any
you said that
questions?
they benefit from the salt, the Staphylococcus?
They don't necessarily benefit from it, but they can grow in 7% sodium chloride. All staphylococcal species can grow in 7% sodium chloride. And if you remember, Staphylococcus epidermidis grows on our skin, which can be up to 20% sodium chloride. Okay?
Okay.
They may grow better on mannitol salt agar than they do on a general purpose media, but they can grow on a general purpose media, like Staphylococcus epidermidis can grow on both the general purpose media and on mannitol salt agar.
Does it grow on the does it grow on skin because of sweat with the salt?
Well, that's where we get our salt on the skin, and that is when we perspire. Salt is put out in the perspiration, and then when it evaporates, it leaves the salt behind. And our skin can be up to 20% sodium chloride. And it depends on the last time you showered or bathed and how much you've been sweating for what your skin will be, the sodium chloride content in your skin will be. Okay, so obviously after you just took a bath, the sodium chloride content of your skin would be very low, like 1% or less. But with time, the sodium chloride content will increase. And the highest it'll get is 20%. And that's just as much sodium chloride as we perspire. We don't perspire higher than 20%. All right, so read through the inoculation, isolation, incubation, inspection, and identification. Inoculation is you just inoculate the bacteria from a clinical specimen on a plate. Isolation. Louis Pasteur developed the concept of pure cultures where he isolated different species from others. However, his procedure was very difficult. He did a limiting dilution in a liquid culture, and he made different dilutions. And if one dilution had only 1 tube in 10 showing growth, he then reasoned that that culture, the 1 in 10, is probably pure. Now, the trouble is you have to make several dilutions, and you only use that one dilution where one bacteria, 1 tube is positive for growth, and 9 tubes there's no growth in them. And you use that to get your pure culture identification. Robert Koch developed a much easier way to get a pure culture. And what you do is you streak the colonies over an agar plate, and you get isolated colonies. And then you take from one isolated colony, and then you streak it on a second agar plate, once again for isolated colonies. And after you pass the procedure through 2 colony isolation events, we say that you probably have a pure culture. If you only do it one time, you may not have a pure culture. And it's true the majority of the times it'll be a pure culture, but you might only, if you only do it once, streaking for isolated colonies, one time in agar plate, you'll probably only have about 70% of the colonies that are one pure species. So you need to streak a second time for isolated colonies on a second agar plate before you can say that you have a pure culture. And even then, it's not 100% guarantee that you will have a pure culture, but it is greater than 95% chance that you will have a pure culture. What you do when you're streaking for colony isolation is you streak in one quadrant, and then streak in another quadrant, then streak in at least a third quadrant, and this one's streaking in 4 quadrants. Someplace along the line, you're hoping to see isolated colonies. And it can be in the 4th quadrant, or it can be in the 3rd quadrant. It doesn't matter where you get the isolated colonies. The point is that when you're streaking for colony isolation, you're putting down individual cells, which can give rise to an isolated colony, meaning one cell can split by division into 1,000,000 or more cells, giving rise to a colony. But if you only do this one time, you don't know, was this colony derived from one cell, or was it derived from 2 cells, or from multiple cells? And so that's why you take an isolated colony and then streak it out a second time on a second agar plate. And when you get the cells through 2 colony isolation events, then we can say that you have a pure culture with greater than 95% chance that it's pure. Any question about any of that? All right. After you put the bacteria on a culture, you then incubate it, and that puts it in a device giving an appropriate environment to culture microbes. There are various physical factors that the incubator can do. Here is an actual incubator. And complicated incubators can provide a specific oxygen environment, a specific CO2 environment. The incubators in our lab only provide a specific temperature and humidity. Some bacteria need humidity in order to grow. And most bacteria have an optimal temperature to grow at. And so our incubator is always warm and provide humidity as well. And obviously we grow them for a specific time in the incubator. Usually we grow them for a day or 2 days in the incubator. But there are other incubators, like this one here has a lot of dials down there. So I'm guessing that you can switch these dials to determine what oxygen that the bacteria can grow under and what CO2. This anaerobic jar removes oxygen from the environment, and you put this in the incubator, and then you can grow anaerobes in the anaerobic jar. And these bacteria will grow if you have no oxygen around to kill the cells. Any question about any of that? All right, come on. You then inspect the cultures to see obvious visible colonies. You can determine certain morphological features from the colony, meaning colonies have specific shapes and different colors, different properties. We'll talk a little bit more about that. And that allows you to differentiate between a few species. However, you should realize that the colonies can take on different properties depending on the age of the colony. Meaning when the colonies are very young, their size will be small. And if they are very old, the colonies can be much bigger. I think that's pretty obvious. But the shapes can change as well. They then identify the bacterium species. And to do that, you generally need to perform staining as well as different biochemical testing. And we'll talk more about that in lab 6 and lab 7 and 9. Staining is what to talk about in lab 4. Any question about that? All right, you should know the terms of this lab. In the laboratory exercises, you should watch a video on inoculation of agar plate using streak plate culture technique. We have a little video on showing you beta hemolysis alpha hemolysis, and gamma hemolysis. And I forgot to mention, you only need to watch this first video until 4 minutes and 15 seconds, because that's the point we're trying to demonstrate what we want you to learn with this video. After that, you can watch it if you want, but we're not going to require it. And then there's a video on McConkey's auger. Only watch it to 3 minutes and 50 seconds. And then a video on mannitol salt auger. And let me take a look at the worksheet and see if there's a virtual lab. It's not in the worksheet. Yeah, there's no virtual lab in the lab anymore. I need to cross that out. So there is no virtual lab in this lab. Where are we? So there is no virtual lab for you to run. Actually, if I would have read that, I would have known that doesn't run because Adobe is no longer supported and won't run on your computer. All right, so answer the questions. And please do not answer it in the lab module. Only answer it in the worksheet. There were a few people who turned in the lab module last time, and I gave you a warning. If those people turn in the lab module again, you're going to get a penalty, a 1-point penalty on the lab. All right, so only turn in the worksheet. The reason for this is the worksheet is much smaller than the lab manual, and I don't want my Canvas web page to fill up. So turn in only the worksheet. So answer the questions. Then fill in this table where you're denoting whether each medium is non-selective, selective, or differential, by placing an X in the appropriate column. And then answer the question, which of these, plate A, B, or C, is using streak plate technique, the colony streak plate technique. And then that's it for the lab. I will be here until 8 o'clock to answer any questions. After that, I think I'm going to go and lie down. All right, so go ahead and get started on the lab. Stop the share. Stop the. I just saw one other thing, and that is for quiz 1, the answers will be available from January 22nd to February 5th. So we can turn that off now.